Functions of three of the basic tissues are simple and straightforward. Muscle tissue contracts. Nervous tissue conducts impulses. Epithelium covers surfaces and/or secretes various products. Connective tissue, however, plays a number of different functional roles. Some of these justify its name by joining together or connecting other body structures, as is readily demonstrated by tendons, ligaments, and joint capsules. Even blood fulfills a connecting role in the philosophical sense by transporting nutrients, wastes, and hormones. Bone tissue and cartilage, however, have clear-cut structural responsibilities, and adipose tissue exists to store energy in the form of fat. Primitive connective tissue, called mesenchyme, doesn't play an active functional role until injury or disease dictates that replacement or reinforcement of existing tissue is warranted.
I'm Mike Shively, and this is Introduction to Anatomy and Physiology. Do any of you have a question from previous session?
I did. I had a question on when we were talking about the three primary germ layers, the undifferentiated cells. What are those, like stem cells, or what are they exactly?
Yeah, when we say, Susan, undifferentiated cells, we're basically talking about cells in the body that, well, for lack of a better illustration, have not decided what they're going to do. These are cells that are held back in reserve that have many differentiation possibilities. They can become a lot of different things. And the body has a number of these back, particularly those tissues that have high regenerative capacity. And when significant portions of tissue, of that particular tissue, like connective tissue and epithelium, are lost, then some of these stem cells can differentiate into the same kinds of cells that are needed to replace that which has been lost. So stem cells fall into that category of undifferentiated cells. You bet. Let's switch to this overhead camera for just a moment, because I've put up here a crude drawing of three different kinds of muscle cells to illustrate something that we talked about last time. This one right here is cylindrical. It has stripes across it. It has, as you can see, multiple nuclei. What kind of a muscle cell would this one be? Anybody?
Skeletal.
All right, Seth, that's a skeletal muscle cell. Here's one that has striations. It's branched. It has a single nucleus. What kind of a muscle cell would that be?
That'd be cardiac.
Right, Susan, that's a cardiac muscle cell. And here's one. I have to say that it's a muscle cell. It's fusiform. That's a little fatter in the middle, and it tapers off or gets thin at the ends. And it has no striations, single nucleus. That's a smooth muscle cell. So that's in the histologic section how these would differ in their appearance. Today, we want to talk about connective tissue and nervous tissue to kind of complete our introduction into the various tissues of the body. Again, we're trying to build a foundation of knowledge so that when we start talking about organs and other structures in the body and say that, well, this particular structure contains this kind of tissue and this kind of tissue, we have some kind of an idea of what we mean by those things. So today we're going to talk about connective tissue and nervous tissue. Do you remember from a previous session when we say connective tissue has an extracellular matrix, what two things are found in the extracellular matrix? Anybody? Susan?
Fibers and ground substance.
Fibers, extracellular fibers made out of protein, and ground substance. Along with the cells, then, there are three components of any connective tissue. Let's look at the first graphic and take a look at what the three components of any connective tissue are. We have cells. Actually, there are several different kinds of cells in connective tissues. We have extracellular fibers, and we have ground substance. Those last two things, which in most connective tissues, the fibers and the ground substance are manufactured by the cells, and those two things comprise what we call the extracellular matrix of that particular connective tissue. Now, the different connective tissues have different kinds of cells in them. They have different kinds of fibers in them, And the ground substance varies from one kind of connective tissue to another kind of connective tissue. In a previous session, we did name or outline what the various kinds of connective tissue were, but I think it would behoove us to review that one more time just to keep it in mind. So let's look at the next graphic, which shows the major types of connective tissue. All of these fall under the heading connective tissue. So we have fibrous connective tissue, also known as connective tissue proper. We have bone tissue, cartilage, blood, adipose tissue, bone marrow tissue, reticular connective tissue, and mesenchyme or primitive connective tissue. So those are the 8 major types of connective tissue. And we want to take these one at a time and talk about some of the features of each one. And of course, we'll begin with connective tissue proper and discuss these in the order that we had them on that particular graphic. Connective tissue proper, also known as fibrous connective tissue, has several different cell types, but the most important one is a fibroblast, called a fibroblast, and it makes the fibers and the ground substance found in the extracellular matrix. Let's think of some of the kinds of things that are formed in the body, primarily of connective tissue proper. From your reading, can you think of something that's made out of CT proper or fibrous connective tissue, at least primarily? I'll start then. A tendon. A tendon, which is part of a muscle, is formed primarily of connective tissue proper. Somebody think of another thing. Seth?
Are ligaments formed of CT proper?
Ligaments. Now, tendons are parts of muscles. Ligaments are structures that actually stabilize joints by basically going from bone to bone. And those are made primarily of connective tissue proper. There are a lot of things in the body that are composed primarily of connective tissue proper. Let's look at a graphic that summarizes these. Tendons, which are parts of muscles, we have mentioned. Ligaments, which stabilize skeletal structures at joints. Fascia, which connects structures together, joins the skin to underlying structures, serves as an avenue of penetration for nerves and blood vessels. Periosteum and endosteum are the names given to the connective tissue proper encasements and linings, respectively, of bones. Epimecium, paramecium, and endomecium are names given to the connective tissue proper encasements around muscles. Epim being around the whole muscle, paramecium being around fibers, a bundle of fibers within the muscle, and endomecium being the encasement of individual muscle cells. The epi, peri, and endoneurium are connective tissue encasements associated with nerves in the same sort of way that the encasements were that we just mentioned in regard to muscle tissue. The meninges are in fact the connective tissue encasements of the brain and spinal cord. Joint capsules, which hold the synovial fluid within synovial joints and also serve to join the skeletal components together in synovial joints, are formed of CT proper. Dermis is the deep of the 2 layers of skin. The outer layer of skin is called, remember, epidermis, and the deeper layer is dermis, and the dermis is actually CT proper. Periodontium, which is the connective tissue that kind of glues the roots of teeth into the bony alveoli which support them. Lamina propria and submucosa down here are connective tissue layers within the intestinal wall. So these are structures in the body that are composed primarily of connective tissue proper. Susan?
When we were talking about dermis, the epidermis would be epithelial cells, wouldn't it?
Yes.
Okay.
Epidermis is stratified squamous epithelium, so it is epithelium. But the dermis, which is the deeper of the 2 layers of skin, is connective tissue proper. So when we talk about skin, we've got epidermis on the outside, and then deeper, we've got dermis, which is CT proper. Any other questions? So, CT proper is very widely distributed in the body, as you can see from the kinds of things that we were talking about. In addition to these, every organ in the body has at least some connective tissue proper within it. So it's extremely widely distributed and forms a physical structural framework for the organs of the body. Now, to understand how it's put together and how it's classified, let's go to the overhead for just a moment. There are 3 criteria primarily related to the extracellular fibers of connective tissue proper, which are used in the classification scheme. The extracellular fibers of CT proper are either loosely packed or they are pretty tightly packed. If those fibers are loosely packed with a lot of space in between them, we say, hey, that's a loose connective tissue. If they're tightly packed to where the fibers are jammed very tightly against each other, we say that's a dense connective tissue. The fibers also are classified as regular or irregular. And what we mean here, regular simply means that the fibers are parallel with each other. They're all running the same direction. And irregular, they're running different directions. Finally, we see terms like collagenous, reticular, and elastic. And these terms refer to the primary protein which is involved in the composition of the fibers. If the fibers are made primarily out of collagen, we refer to that fiber as a collagenous fiber. If the protein involved is reticulin, we call that a reticular fiber. And if the protein involved is elastin, we call that an elastic fiber. Now, all of these fibers, all 3 kinds are actually manufactured by the primary cell, again, we mentioned this a while ago, but I want to put it up here again, of connective tissue proper, which is called a fibroblast. So the fibroblasts make all 3 of these kinds of fibers. If after they extrude them extracellularly, they are parallel with each other, we use the term regular. If they're going different directions, we use the term irregular. If they are tightly packed, we say dense. If they're loosely packed, we say loose. Now let me show you how that is used in a classification. If we were to talk about, say, a tendon or a ligament, we would say that is composed primarily of dense, regular, collagenous connective tissue proper. And so the word dense tells us that the fibers are what? Tightly packed. When we use the word regular, hey, that's talking about fibers being parallel with each other. They're all going more or less the same direction. When we use the word collagenous, the implication is, hey, most of these fibers are made out of the protein collagen. Now, that doesn't mean there can't be some elastic fibers in there or reticular fibers, but it talks about when we use one of those terms like elastic, reticular, or collagenous, that implies the composition of the predominant fibers. Starley?
If it's regular or irregular, does that cause striation at all? Like if they're regular,
no, it doesn't. The striations that we talked about in regard to muscle tissue have to do with the way that the contractile proteins are arranged, but they're always parallel in muscle cells, even in the muscle cells, the smooth muscle cells that are not striated. So we don't get any striations based on the loose or dense or the regular or irregular nature of the arrangement. Any other questions? Let's give you another example. Superficial fascia, which is the connective tissue proper that glues the skin down to bones and muscles and that sort of thing. That is loose, irregular, collagenous connective tissue proper. Loose meaning, hey, the fibers are not tightly packed. irregular, meaning the fibers are going different direction. Again, collagenous, implying that the predominant fibers are made out of the protein collagen. And then connective tissue proper, telling us that, hey, this belongs to this first category, fibrous connective tissue that we're talking about. Do those concepts of classification make sense to you? It's a relatively simple concept. Okay, let's take a look at a graphic and make sure that we've got all those words down in case you couldn't read my writing here on the overhead. Again, loose and dense refer to how tightly packed the fibers are. Regular and irregular refer to whether the fibers are parallel or not, and the other words there refer to the predominant fiber type, the predominant protein making up the fibers that are present. So that's a thumbnail sketch of connective tissue proper. Now let's go to the next of the connective tissues. We'll talk a bit about bone tissue. Let's go to the overhead for just a moment. In bone tissue, we have three different kinds of cells. The beginning prefix of all these is the same. It's osteo. And the cells that we call osteoblasts, these are bone-forming cells. The cells that we call osteocytes are kind of sedentary cells that aren't doing much at the time. They're old osteoblasts, so once osteoblasts get done with their particular part of forming bone tissue, they become osteocytes. And finally, we have cells that are called osteoclasts. And these are bone tissue reabsorbing cells. So blast means forming. These are bone-forming cells. These are simply bone cells that sit in the tissue once it has been formed. And these are bone tissue reabsorbing cells, osteoclasts. These are multinucleated, these last ones. And they probably are able to reabsorb bone tissue because they actually secrete an acid which dissolves the mineral component of bone tissue, and then they secrete enzymes which actually break down the fibrous portion, and thus the bone tissue can slowly but surely be reabsorbed. Starley?
Sorry, well, osteoblasts are forming it. Why would it want to be destroyed? Why does it keep going in a cycle?
It's an interesting concept. Obviously, when we are growing as children, the osteoblastic activity or bone tissue formation activity predominates. But even while we are growing, there is some osteoclastic activity. Bones are in what we call a dynamic state all the time. Bone tissue is being laid down and bone tissue is being reabsorbed. But while we're growing, more bone tissue is being laid down or manufactured than is being reabsorbed. Throughout most of our adult life, we're kind of in a status quo with about the same amount of absorption as reabsorption as bone tissue formation. Finally, when we enter senility, there's actually a slight excess of bone tissue reabsorption. And that's the reason, as an older person, it's so easy, say, to fall down and break something, because the bones simply aren't as strong. Some of the bone tissue has been reabsorbed in old age. So the reabsorption exceeds the formation in senility. Now, let's go back to this overhead for just a second. Osteoblasts make both the fibers and the ground substance of bone tissue. I'm just going to write G.S for ground substance. The fibers are very heavy collagenous fibers. And the ground substance, again, is a protein polysaccharide, a little different than the protein polysaccharide found in other ground substances, but nonetheless the same idea, kind of a glue. So osteoblasts make the fibers, they make the ground substance. Once the fibers are manufactured, then calcium phosphate salts are actually deposited on the surface of the fibers. So we get complex forms of calcium phosphate, that actually come in here and are deposited on the surface of the collagenous fibers. And that is what makes bone tissue so hard. In other words, the mineral content is not found within the cells, because it would, of course, kill them, but it's deposited on the fibers. Now, we take a bone out of a person or an animal and put it in an acid material which will dissolve the mineral, then that bone can be, it becomes extremely flexible, just like a piece of rope. You know, you can take a femur bone, once you've demineralized it, and you can actually tie it up into a knot, you know, just like you're tying a bow. So it's the mineral that makes it actually so very stiff. And bone tissue is extremely, extremely hard. Matter of fact, there have been some studies where they've taken similar quantities of bone tissue and oak, the wood oak, which is a very strong wood, and on a per quantity basis, bone tissue is actually physically stronger than oak. Did you have a question? Okay. Let's go back to this overhead one more time. Now, when the osteoblasts secrete the fibers and ground substance, some of them become so enthusiastic about manufacturing this extracellular matrix that they kind of paint themselves into a corner and get caught within a little pocket of the extracellular matrix that they are producing. And so when we look at bone tissue underneath the microscope, we'll actually see within the extracellular matrix little tiny cavities called lacunae, and inside each one of these lacunae there will actually be a bone cell. Once one of these osteoblasts gets trapped in one of these lacunae, it then becomes an osteocyte. So basically, while the cell is up here actually producing the fibers and ground substance, we're going to call it an osteoblast. As soon as that particular osteoblast gets entrapped because it paints itself into a corner, so to speak, and is now conducting a hermetic type existence in one of these lacunae, we're going to call it an osteocyte. And so when you look at bone tissue, you see a lot, underneath the microscope, you see a lot of little isolated pockets. Each one of those little pockets then contains one osteocyte. Yeah, Bryce?
So does the osteoblast die and then stay in that position, or does it keep
No, the
producing?
osteoblast just simply becomes, by definition, an osteocyte. They stay alive. And we're not sure what osteocytes do in a lacunae. There's probably not a whole lot to do in there. But they certainly have a very, very low metabolic activity. Any other questions on that? Now, let's go back to the overhead yet one more time. Let's assume that we draw a circle here, put a little thickness on it, and say this is a transverse section through our humerus when we're, say, 6 years old. And then we'll draw a much larger circle down here and say this is a transverse section through our humerus as an adult. And of course, not only has the humerus grown in overall diameter from the time we were 6 until we're an adult, but also the medullary cavity in here has enlarged. So in reference to one of the questions that was asked a little bit ago, even while a bone is growing, we're going to have a lot of osteoclastic activity, because to go from here to here, we must have bone tissue laid down on the outside, similar to the growth rings in a tree, in order to enlarge the cortex and make it bigger in diameter out there. At the same time, there has to be a lot of bone reabsorption going along here in the medullary cavity side in order to enlarge the medullary cavity. So we have osteoblastic activity out on the outside, and we have osteoclastic activity on the inside as a bone grows in diameter. That make logical sense? Yeah, Starling.
Where are osteoblasts created? I know osteocytes come from osteoblasts.
During differentiation, very early on, when we talked about all the connective tissues coming from mesoderm, so we got this primary germ layer called mesoderm. It develops into all of the different kinds of connective tissue. So some of those cells differentiate into osteoblasts, which start forming bone tissue. Some of those other cells form the osteoclasts, and so forth. Okay, so it all comes from mesoderm originally.
So when the osteoblasts keep getting stuck in the lacunae, then don't you get fewer numbers of osteoblasts? And is that why the osteoclasts overtake it in senility?
We believe that osteoblasts themselves can either divide, or stem-type cells that form osteoblasts continue to form new osteoblasts, because you're exactly right. Osteoblasts are continually being lost because they paint themselves into these lacunae and then by definition become osteocytes. And once they're in a lacuna, they're done with the bone tissue formation. They're lost as far as bone tissue formation, so they will just sit there in a relatively low metabolic rate for the life of the individual. Exactly right. Any other questions? Yeah, Seth?
Yeah, you mentioned in an earlier session that red blood cells are created in the medullary cavity of bones. Is that in all bones?
Essentially, all bones during development have some bone marrow tissue that is hemopoietic or bone cell forming. There probably are a few bones, like the little teeny tiny auditory ossicles. I don't think there's enough, bone marrow tissue within them to talk about any red blood cell formation. But most bones have that, and of course, when the bones are younger, we have a lot more of that going on than we do in an adult. But yeah, most bones have some hemopoietic activity or blood cell forming capability, at least sometime during their development. Yeah.
So does the strength of the bone, is it affected when children are younger than when they're older? Does its durability change?
That's a good question, Bryce. In general, bones of children aren't quite as well mineralized. They're not as big, so a given force is more likely to break a child's bone because it's simply not as massive as that of an adult. Doesn't probably have quite, even on a per mass basis, doesn't have quite as high a mineral content. And I think I mentioned this once before in one of our earlier sessions, but when astronauts go into a weightless environment in outer space and spend extended periods in outer space, say at the space station, during that period of time with less stresses and strains on the skeletal system, there will be some demineralization and the bones will weaken somewhat. Now, they don't typically weaken enough that these guys get off the shuttle and fall down and break something, but nonetheless there is some demineralization that occurs. Any other questions? Amber?
And babies, like they're more kind of cartilaginous, like their bones are more flimsy, or you know what I mean, like they're not as hard. Is that still the same stuff as the bones?
That's a good question. When we talk about the skeletal system in a future session, we're actually going to talk a little bit about bone development, but a significant percentage of the bones in the skeleton start out in a cartilaginous state and then undergo a mineralization, actually an ossification process that converts those cartilaginous models or precursors into bone tissue. Yeah, sure happens. There are a few bones that don't have that cartilaginous state, but most of them do. Starley?
I just was wondering, when osteoclasts are reabsorbing and they hit a lacuna, does it actually free the osteocyte and they become an osteoblast again?
That's an excellent question, and I don't know that I've ever read the answer to that, but when you think about what they have to secrete in order to demineralize, it has to be a very low pH secretion to break the mineral down. And then there's some really powerful enzymes that are breaking the collagen fibers down. I think the osteocytes are probably killed rather than freed, to be honest with you, when an osteoclast is reabsorbing. Yeah.
Thank you.
Good questions. Let's talk a bit about cartilage. Now, cartilage is different than bone tissue, and it's not just because it doesn't have mineral in it. Okay? So I don't want you to think that if we have unmineralized bone tissue that it's cartilage, because that's simply not the case. The cells are different, although they have similar names. There's different fibers present in cartilage, and the ground substance is slightly different than that found within bone tissue. Now, we said that the three kinds of bone cells were osteoblasts, osteocytes, and osteoclasts. In case you couldn't read my writing, let's look at the graphic that shows those. Osteoblasts form bone tissue. Osteocytes are the ones that sit around in the lacuna after the bone tissue has been formed, and osteoclasts are the multinucleated bone tissue reabsorbing cells. If we look at the next graphic, we can see that the cells of cartilage are named in a similar manner. The cartilage forming cells are called chondroblasts. The cells that sit around in the cartilage matrix after it's been formed are called chondrocytes. And some histologists refer to the multinucleated cartilage reabsorbing cells as chondroclasts. And the reason I say some histologists, because there are those histologists that prefer to call these, that say that they're really just osteoblasts that are doing that. But I don't really want to debate that unsettled issue. So we've got three kinds of cells that are parallel in name and in function to those found in bone tissue. Now, when we think about the kinds of fibers, I said the fibers were different. In bone tissue, we said they're large collagenous fibers. Well, chondroblasts actually make three different kinds of fibers. So if we'll go to next graphic, you'll see what these fibers are. The chondroblasts that produce primarily collagenous fibers, that kind of cartilage is going to be called fibrocartilage. Other chondroblasts produce primarily elastic fibers made out of elastin, and that kind of cartilage is going to be called elastic cartilage. And finally, the chondroblasts that produce what are called reticular fibers, they're almost the same as the reticular fibers that we talked about in connective tissue proper. That kind of cartilage is going to be called hyaline cartilage. So we actually have then these three different kinds of cartilage. We have fibrocartilage, elastic cartilage, and hyaline cartilage. We only got one kind of bone tissue, but we have three kinds of cartilage. And the kind of cartilage is dependent on the predominant fiber type in the extracellular matrix. Now, the three kinds of cartilage are variously distributed in the body. We have in, say, the articular surfaces of bones, where bones come up against each other to form synovial joints, that kind of cartilage is hyaline. And the kind of cartilage that forms the cartilaginous extensions of the ribs coming on around from the bony part of the ribs back here, that is hyaline cartilage. The kind of cartilage that we find primarily in the cartilaginous structural framework within the larynx, there's quite a bit of elastic cartilage in there. And there's elastic cartilage forming the stiffening of the external ear. So there's some flexibility here because of the elastic fibers. Thus, when you bend your ear around, you don't break it off. You know, that'd be inconvenient, to say the least. And then fibrocartilage, the third type, is found in places like in the intervertebral discs between adjacent vertebrae within the vertebral column. That cartilage that forms those discs is made of fibrocartilage. So we'll take a look at a graphic that kind of summarizes what we just said there. Hyaline cartilage, here's some of the kinds of places. Articular cartilages, tracheal cartilages, which form the stiffeners within the trachea. If we didn't have those, what would happen when we inhaled? Our trachea would tend to collapse, wouldn't it? Just like the vacuum cleaner hose has some stiffeners in its wall, same idea. The costal cartilages we mentioned, some laryngeal cartilages are hyaline, some are elastic. The nasal cartilages tend to be hyaline cartilage. And the epiphyseal cartilage, which are sometimes called the growth plates in growing bones, in growing long bones, are made of hyaline cartilage. And then other structures made of fibrocartilage, we mentioned intervertebral discs, but some structures that are similar in structure called articular discs, which are found in some synovial joints, and articular menisci are made of fibrocartilage. The interpubic disc, which we'll take a look at in a future session on a skeletal model, is made of fibrocartilage. And then the glenoid labrum and acetabular labrum that's formed of fibrocartilage, and that kind of deepens the acetabulum and the glenoid fossa of the scapulae. Did you have a question, Starley?
Oh, sorry. I was just wondering, how does it know which kind of fiber to make?
You know, we don't know a lot about how. There's obviously internal chemical regulators during growth that determine these kinds of things, but a lot of that is not clearly understood at all. Obviously, you know, there has to be, because it turns out that same kind of stuff is found in the same place in different people, you know, so it's not a random kind of thing. Good question, don't know the answer to that one. Okay, back to this same graphic now, structures composed of cartilage. The elastic cartilage is found in some laryngeal cartilages we mentioned. There's some in certain of the nasal cartilages as well. So your nose has a little bit of flexibility down at the distal part of the nose, right? Any questions about cartilage? All right, let's talk a bit about blood. And let's go to the overhead for a minute. Blood can be divided into what we call plasma and formed elements. This is the liquid part, and this is where we have the cells. Matter of fact, the formed elements can be divided into 3 categories: RBCs, red blood corpuscles, also known as erythrocytes; WBCs, white blood cells, also known as leukocytes; and platelets, also known as thrombocytes. So we've got these 3 kinds of formed elements. And actually, there's 5 kinds of these. And we'll talk about that more when we get to the cardiovascular system. Of course, the red blood corpuscles are involved in carrying oxygen because of their hemoglobin content. White blood cells, or leukocytes, have various functions, but in general, they have defensive roles. And platelets are involved primarily in the clotting mechanism. The formed elements actually make up about 45% of the blood volume. And then the plasma over here is about 55%. The plasma itself is mostly water, but there are some proteins and other organic materials in it. So when we talk about blood, just separate in your mind the concept of plasma versus formed elements. Yeah, Starley?
I know when people donate plasma, are they donating water then? Or
It's interesting that you should say that. I happen to be a plasma donor myself, and plasma is, in fact, about 91 or 2%. It's roughly 90% water. So when you donate plasma, you're donating primarily water. In the plasmapheresis machines that they use, they actually they pull out a certain amount of blood, spin it, and centrifuge the cells away from the liquid part, draw the liquid part off in the collection container, and then infuse your cells back into you. So you basically don't lose cells whenever you're donating plasma. That's one of the reasons why plasma donors can donate twice a week with regularity, whereas blood donors, they can only donate every few weeks because it takes much longer time to replace the lost blood cells. Yeah, Amber?
So when you donate plasma versus when donating blood, does it matter like the blood type that you have for the receiver, the person who's receiving it?
Well, we'll talk about transfusion reactions a little bit when we talk about the cardiovascular system, but in general, the type of blood that you have is based on some components of the red blood corpuscles and has basically nothing to do with the plasma component. So, yeah, Bryce?
Do some infections or diseases like HIV infect the plasma? Or is
Oh,
that just
yes.
in the...
As a matter of fact, the plasma donors are very carefully screened for HIV and some other diseases as well. All right. Let's take a look at what the functions. Well, first of all, let's see if we can summarize. Let's look at a graphic that'll show you again what we just said as far as the components of blood are concerned. Again, formed elements about 45%, erythrocytes, leukocytes, and platelets, and plasma about 55%. And again, over 90% of that plasma, over 90% of the 55% is water. There are several different kinds of proteins, albumins, globulins, and even fibrinogen, which is involved in the clotting process. And then we've got a few other organic compounds and, of course, a few inorganic compounds as well found in the plasma. Now, what does blood do? Let's just talk theoretically among ourselves before we take a look at a graphic that'll summarize it. What kinds of things, why do we have blood circulating in our vessels? Yeah, Starley?
It moves oxygen throughout the body.
Going to transport oxygen, going to transport carbon dioxide, going to transport other nutrients, going to transport other wastes. It's a great transport medium, is it not? Hormones are transported through the bloodstream, so there's a lot of transportation going on. Anything else? Does it have any defensive roles? Anything about blood defensive as far as the body in combating disease?
Doesn't
Susan?
it carry the white blood cells that have the immune system?
We've got white blood cells that produce gamma globulins or produce antibodies. We've also got the clotting mechanism, which is defensive. We've also got phagocytosis of certain blood cells, okay? And blood helps maintain pH. It's one of 3 components of the body that have a role in maintaining a steady state as far as pH is concerned. And certainly, it helps maintain temperature. Yeah. Did you have one, Tyler, above and beyond?
Temperature.
I got it. All right. Let's take a look at a graphic that summarizes these now. Transports nutrients, wastes, and hormones. Has several different defensive roles that we mentioned. It helps maintain body pH, and it's involved in thermoregulation. Now, we've talked a little bit about connective tissue proper. We've talked a little bit about bone tissue, a little bit about cartilage, and a little bit about blood. Now, I want to just very quickly look at the other connective tissues. And so we'll look at a graphic that summarizes the main points that we would have made. Adipose tissue, which, as we mentioned before, is commonly called fat, is really just connective tissue proper with a very large population of adipose sites or fat cells within it. Reticular connective tissue, we have some modified fibroblastic type cells that are called reticulocytes. They form reticular fibers, which in turn form the structural framework within many of the lymphatic organs. Bone marrow tissue, we've mentioned, is found in medullary cavities of bones and is hemopoietic, or has the precursors of various blood cells within it. And finally, mesenchyme is primitive connective tissue containing a lot of, quote, stem cells. So those are the other kinds of connective tissues, and I just wanted to briefly mention them. I don't want to spend a great deal of time talking about them because we need to move on to nervous tissue. Do you have a question, Starley?
I was just wondering what streaking was caused by. My sister had stepped on a tack, and then her veins started getting really dark up her foot.
The most common cause of streaking in regard to a puncture wound or some kind of a traumatic injury is when lymphatic vessels that drain the area injured get inflamed, perhaps with there may be microbial cells involved in that process, but it's called lymphangitis, and the vessels begin to show up as commonly kind of a dark red streaking. And that just implies that we've got an infectious process that's channeling its way up the lymphatic vessels. Bryce?
So is that something that your body will usually take care of, or is it?
Sometimes it is, and sometimes we need help. It's usually pretty dire circumstances. When you see that, you probably ought to seek medical attention right away, and they'll probably put you on a pretty strict regimen of antibiotic therapy.
I just have a quick question on bone marrow transplants. why do they do the bone marrow transplants? It's the body not able to make the red blood cells, or what happens
Sometimes,
with those?
Susan, we have some of the white blood cell precursors that have gone neoplastic, and they may use chemotherapy or radiation to wipe out those cells that are also formed in the bone marrow tissue, and then to replace those disease cells with healthy cells, they'll do a transplant and get a new population of cells started. Does that make sense? Mm-hmm. Yeah. Let's talk about, in our remaining time, let's talk a little bit about nervous tissue. And we don't have a whole lot to say because, as you recall, there are no subtypes of nervous tissue. You remember the function is to conduct nervous impulses. But there are two different populations of cells within nervous tissue. The ones that we call neurons do the impulse conduction, and the supporting cells are called glial cells or neuroglia. Let's look at a graphic that just summarizes this. There are three different kinds of neurons. Neurons that we call multipolar, ones that we call bipolar, and ones that we call pseudo-unipolar. And there's 6 different kinds of glial cells or supporting cells. Remember, it's just the neurons that do the impulse conduction, and these support the neurons in various physical and metabolic ways. Let's go back to this classification, and let's go to our overhead camera, and I'll show you the difference between what we mean by multipolar, bipolar, and pseudo-unipolar neurons. If we're very, very schematic, and we say this is the cell body of the neuron, there's its nucleus, there's the process that we're going to call dendrite, here's process that we're going to call axon, then this is considered to be a bipolar neuron. Has one dendrite and one axon. There are not very many of those neurons in the body. If, on the other hand, the cell body is offset, we could call this dendrite and this axon. This is a pseudo-unipolar. Neuron. There are a lot of these in the body, and there are also a lot of neurons that have this configuration. Multiple dendrites, single axon. And this is the configuration of a multipolar neuron. Now, multipolar neurons are the kind that carry impulses out from the brain and spinal cord to other body structures. They are motor neurons, multipolar and motor, go more or less together. Pseudo-unipolar neurons form most of the sensory neurons in the body. And sensory, by sensory I mean conducting impulses into the brain or spinal cord. So when you think of multipolar, think motor. When you think of pseudo-unipolar, think sensory. And the bipolar neurons, we said, were not very common in the body, but they are also sensory where they occur. And again, just to make sure we're on the same wavelength here, when we say motor, the implication here is the direction of impulse conduction is out, away from the brain or spinal cord. Starts in the brain or spinal cord and goes out to the peripheral part of the body. When we say sensory, the implication is impulses are being conducted in these neurons into the brain or spinal cord. And I want to make sure that you understand at this point, it's not the same neurons that are doing this at different times. A given neuron is either a sensory neuron or a motor neuron. And a lot of people are kind of surprised at that. They maybe figure that neurons are two-way streets for nervous impulses, and they're really not. A given neuron is either a motor neuron or it's a sensory neuron. Does that make sense? So it's different neurons. We have different ones for different jobs. They're very specialized. They're one-way streets as far as impulse conduction is concerned. Now, another thing that you might not have thought about, when a neuron gets out to wherever it's going with its axon. That next cell that it interacts with can't be just any cell in the body. There's only three real possibilities. It's either another neuron, or it's a muscle cell, or it's a glandular epithelial cell. Those are the only three kinds of cells with which a given neuron can interact. Neurons never interact with blood cells, connective tissue cells, membranous epithelial cells, you see. They always synapse with, and when we say synapse, we're talking about the junction of a neuron with another cell. That's a synapse, junction of a neuron with another cell. They always synapse with muscle cells, glandular epithelial cells, or with other neurons. Starling?
Can you explain the axon dendrite, which one's going in or out?
Excellent. I should have done that before. Let's go back to our overhead just a minute, and we'll just draw the first neuron that we put up again. If this is the normal direction, physiologic direction of impulse conduction, as indicated by the arrow, the process over here leading up to the main part of the cell where the nucleus is located, this is dendrite by definition. And this part over here, which carries the impulse on away from the cell body out to wherever it's going, is called axon. And as you notice from the three types of neurons that we drew, all neurons have a single axon. Although it may branch out here at the end, it's still just one axon. Pseudo-unipolar and bipolar neurons have a single dendrite. Multipolar neurons have multiple dendrites.
Thank you.
You bet. Now, out here at the tip of the axon, there are chemicals that are released. Do any of you remember what we call those chemicals that are released at the end of an axon?
Acetylcholine.
Oh, you're thinking of specific, the general name. I'm looking for the general name for what happens. We get out here at the end of the axon, the impulse comes out, and then the chemical is called...
Are they just neurotransmitters?
Neurotransmitter, thanks, Seth. So what happens at the end of the axon is that there's a chemical called a neurotransmitter that's released. It diffuses across the synaptic gap or cleft, and it interacts physically with the cell membrane of the next cell. And then, depending on which neurotransmitter is released, that may cause that next cell to do whatever it does or not to do whatever it's supposed to do. There are excitatory type neurotransmitters, and there are inhibitory type neurotransmitters as well. So that's a quick look at neurons. Let's look quickly at the kinds of glial cells or support cells, neuroglia. Let's go to the overhead camera. And take a look at what we're talking about here. These are cells that do not conduct impulses. So none of these conduct impulses. There's actually 6 different types. One has been called for years Schwann cells. A more modern name for this, although you'll still see this in most textbooks, is neurolimocyte. Second one is oligodendrocyte, also called oligodendroglia. And then we've got astrocytes, microglia, ependymal cells, and finally, satellite cells. Now let's start at the bottom and work backwards as we talk about what these do. These support the neurons that are found in ganglia. They're just physically and metabolically supportive of ganglion-type neurons. And we'll talk about what ganglia are later on when we talk about the nervous system. These cells line the fluid-filled spaces within the brain and the spinal cord. They line the central canal of the spinal cord, and they line the ventricles of the brain. These are phagocytic cells that are involved in a defensive role within the central nervous system. These astrocytes form what's called the blood-brain barrier. And it turns out that things that are floating through the bloodstream, when they diffuse out of the capillaries into nervous tissue, before they can get to the cells within the brain, they have to go through astrocytes. The top two, Schwann cells or neurolimocytes, and the oligodendrocytes or oligodendroglia, these two are both involved in forming what's called myelin ensheathments around the axons of neurons. And the reason we got two different kinds of cells here is because the Schwann cells, the number one that we had on our list there, they do that myelin ensheathment in the peripheral nervous system, and the others, the oligodendroglia, form the myelin sheaths within the central nervous system. Now, a myelin sheath consists of specialized cells that wrap themselves around the axons of neurons. And by doing so, they not only insulate that axon, but they also speed up the impulse transmission. If a neuron has this myelin ensheathment, that neuron is said to be myelinated. And most of the axons of neurons in both the peripheral nervous system and central nervous system do have this myelin ensheathment, which speeds up the impulse transmission to a tremendous degree. But please appreciate that impulse transmission is pretty fast even without the myelin ensheathment. It just makes it a little bit faster. But, you know, if you're stepping on a thumbtack, or as a grandfather, if you're barefoot on a Duplo block in a dark bathroom, then you appreciate the greater speed of impulse transmission because it helps get that information processed and get you off that block just a little bit faster. Helps our reaction time a bit. Does that make sense to you? Are there any other questions? Starley?
I was just wondering, when it's myelinated, how does that speed up the process?
That's a good question. We'll actually explain that in more detail when we talk about the nervous system and impulse conduction. But basically, it's like if you and I are racing up a stairway and I say, you have to take every step, but I'm going to skip steps if I want. I can simply go faster, and it's actually a skipping type of impulse transmission that does that.